All right, welcome to our webinar today, everyone. My name is Ron Myers. I'm the Director of Editorial here at JoVE. So today we'll be having a webinar hosted by Amanda Barrasso. Um, her group published with us in 2018, and she published a technique on mesenchymal stem cells. So she's going to be updating us on the technique today, talking about different variations of the technique and recent developments. So we will be recording the webinar, and we'll send the recording around to everyone after, in the next day or two. And we'll also be having a Q&A session at the end. So feel free to add your comments in the Q&A, and we will address those towards the end. All right. Well, I will give it to Amanda. Go ahead.
Okay, thank you very much for the introduction. Good morning, everyone, and thank you all for joining in today. I would like to acknowledge JoVE for the kind invitation. It's a pleasure to be here talking about this specifically paper. So in 2018, we published with JoVE the paper entitled Derivation and Differentiation of Canine Ovarian Mesenchymal Stem Cells, and that's what I'll be showing you today. So the goals of this presentation is to show the isolation, the expansion, and differentiation of mesenchymal stem cells derived from the canine ovarian tissue, just as published in JoVE in 2018, to show some innovations in the technique that we have done throughout those years, and also some clinical applications, just to show why it's so important to do this technique. So let's start talking about the mesenchymal stem cell isolation. And you may ask, why ovarian stem cells? Well, all over the planet, thousands of female dogs are spayed every day, and the ovary is considered biological waste. However, we have shown that this biological waste can be used to easily isolate mesenchymal stem cells and be transformed in a tool that can be used in veterinary regenerative medicine for a diverse variety of animal treatments. So let's go through the protocol a little bit. After castration, the ovaries, they should be transferred into a sterile tube with PBS, which should be kept on ice until arrival in the lab. In order to wash the ovaries, they should be transferred into a Petri dish with PBS as well. Next, the tissue should be minced into small pieces, approximately 1 millimeter in size. And in order to liberate the cells from the tissue, the collagen bonds need to be break down, which we do by the incubation with collagenase for 3 hours at 37 degrees. And there are a lot of ways that we can extract those cells from the tissue. So we use the collagenase, as I said before. It's one of the most used methods, the enzymatic one, in which we use a proteolytic enzyme to digest the tissue. In this protocol in particular, as I said, we use collagenase to cleave the collagen bonds, and then the cells are released from the tissue matrix. Okay, so what do we do after that? There are different ways to go from here when we are isolating stem cells from the tissue. You can either use a cell sorting machine to select the cells with the mesenchymal markers. You can do the selection by cell sorting. Even though it's a good technique, the drawback is that it's a pretty expensive machine. The antibodies are not cheap either. It can be harmful to the cells, so we don't isolate we don't get a lot of healthy cells in the end. And it's not easy to find a cell sort. It's not everybody that has a cell sort in the lab. So in order to overcome this issue, we have described this method that is very simple, that is inexpensive to isolate the mesenchymal stem cells that I'm going to show you right now. So this methodology is a lot more feasible because there's no need for expensive equipment, and we can recover a large amount of healthy cells after the protocol. So we talked about the steps that we should do until the collagenase incubation, which should be done at 37 degrees for 3 hours. And then after that, we should centrifuge the tube. After these 3 hours, we should centrifuge the tube at 700 G for 15 minutes. After the centrifugation, the pellet should be transferred into a 25 bottle, a T25 bottle, and incubate for 3 hours. And this is the most important step, because after this period, the media should be changed. And this will select those cells that adhere most rapidly to the plastic and consequently resulting in a homogeneous population of mesenchymal stem cells. So usually protocols to recover MSC from the tissue are based on adherence during first 48 to 72 hours of culture. However, it can lead to a heterogeneous population of cells. So after this 3 hours, we can change the media, and the media should be changed for the first time, and then the cells can be expanded, stored, and used for characterization and future experiments. So following 48 hours of culture, we can see the mesenchymal stem cells will start growing, and due to this 3-hour selection procedure, the result in the population is more homogeneous, as we can see in this figure. So here we see cells reaching confluence, and we can observe the fibroblastic-like morphology. And the cells also display a typical MSC molecular profile, as we can see here. So the cells express transcripts for mesenchymal markers, CD90, CD44, CD105, and there are also negative for 2 hematopoietic markers, CD34 and CD45, which is expected. In regards to their differentiation ability, the ovarian MSC have a wide differentiation capability. As we can see in this figure, they successfully underwent not only the mesodermal lineage, as we can see here, such as osteogenic, adipogenic, and chondrogenic lineage, but also successfully differentiate into neurogenic, endodermal precursor, and even primordial germ cell lineages. Okay, so now that we talked about the first protocol that we came up to isolate mesenchymal stem cells from the ovaries, now I would like to share some innovation that have been made to the technique. So first innovation is that we had applied this technique in different species, always with a positive result. And then throughout the years, we have made some adjustments that I'm going to show right now. So here we can see a porcine ovarian tissue, which was minced just like I showed for the dog ovary. However, the incubation with collagenase was also made at 37 degrees. However, it was made with a magnetic stir. at 1,000 RPM in the incubator at also 37 degrees. But instead of doing that for 3 hours, we did that for 1 hour. So we reduced the time of the total protocol, and we had a better result because we have more digestion. The tissue was more digest with the magnetic stir. So after this 1 hour of incubation with the collagenase in agitation, the protocol was the same and the cells were incubated in the T25 bottle for 3 hours, and the media was also changed after those 3 hours. And we had the same results. So, which is great. So this adjustment in the technique just made the protocol a little shorter. And we also could isolate more cells from the tissues due to the fact that the tissue was more effectively digested. So just to finalize, I would like to share with you why it's so important to use this biological waste that is the ovary and transform as a regenerative tool. So here I show one of the clinical application. This is a dog, a patient that we have with corneal ulcer, classified as a surgical emergency, and the cells were injected. And we can see here in figure B that 72 hours after one of the eyes was completely healed, the other eye that had a bigger wound was not healed yet, but throughout the time, it was completely healed. So after 30 days, we can see that both eyes were healed. We had no wound anymore in the cornea. And after 60 days, no scar tissue was observed. So after only one ovarian MS injection and no surgical intervention, we could cure this dog. So it was pretty good, and it's a pretty interesting regenerative tool that we can use in the veterinary medicine. So just to conclude, here we show the isolation of a homogeneous MSC population based on cell adherence to the plastic during the first 3 hours of culture without the necessity of cell sorting, representing an inexpensive, streamlined, and effective procedure. So those are the references. I would like to thank you all for listening, and I'm very happy to answer any questions you may have.
All right, excellent. Thank you, Amanda. And as you mentioned, yeah, if anyone has any questions, just add those to the Q&A. And also we will be, as I mentioned before, we will be sending the recording around. So if if you don't have any questions now and you think of questions later, you can certainly ask them during the email campaign, and we can discuss then as well.
Yeah, sure. I would also be happy to answer any questions later. I put my email here.
Okay, so we just had one question come up. How long were you able to passage
How
the cells?
long?
Sorry, go ahead.
Yeah, no, it's fine. I was just reading the question. So I was able to pass those cells until passage 25. And then they start reaching senescence.
Okay, thank you for the question. Okay, and it looks like someone is raising their hand. I don't think we have that activated, but if you could just type your question in the Q&A.
So I can see here another question from Laura. Thank you, Laura, for your question. Have you tried this methodology with weakling ovaries? So no, I have never tried with weakling. That's a very interesting question because I have tried with cats, with pigs' ovaries, with cows, even with capybaras, and but I have never done with horses. So that would be really interesting to try. Thank you very much for your question. So we have one more question here. What type of collagenase did I use? So thank you for the question. I use collagenase type 1. I have one more question here. If I could modify the cells genetically. So yes, we can. Those cells, they grow as a monolayer, so you can treat them afterwards for whatever you want to do in your experiment. Me, for example, I'm doing a postdoc at NCSU, and I can modify those cells with CRISPR-Cas. So yeah, you can do that. We have another question here. If it's possible to use this protocol to isolate from adipose tissue? Yes, it is. In fact, in this paper that we published, everything that we did for the ovarian tissue, we used the adipose tissue as a control. So we did this with adipose tissue, and we have great results as well. Thank you for your question. And yes, for all these other species that I said, I used this methodology with great success. We have one more question here. At what stage cell number I used for corneal ulcers? So we used 2 million cells, if I'm not mistaken. I can check in that paper, but if I'm not mistaken, we used 2 million cells. Someone else asked here if I use antibiotics on top of collagenase. Usually I don't. You can add 1%, but usually I don't. I add the antibiotic in my culture media. So I usually use DMM with 10% of fetal serum, bovine fetal serum, and then I add 1% of antibiotics. So Laura has another question. Do you think that these ovarian cells may be more pluripotent than the other cells of MSC based on the observations on proliferation and differentiation? This is a very interesting question. Yes, I think so. And that's why we also did the transcripts for some pluripotent markers such as MANOG and OCT4, SOX2, and we did observe the expression of OCT4 that was higher than the adipose tissue. So yeah, we believe that it can be more pluripotent source of MSCs in the adult body. So we have one more question here. How much the percentage of isolated cells? I'm sorry, but I don't really understand that question. We have the cells number that it was viable after, if that was your question. We have that in the paper. I don't remember right now, but usually after 48 hours, the cells are good to be passed. In a T25 bottle, we can start passaging them after 48 hours usually. Do you know what cell density should be reached for extracellular vesicles isolation? No, I'm sorry. We didn't do extracellular vesicles isolation in this protocol, so I cannot tell you that. I'm sorry. One more question. If they are more pluripotent, do you think that it can be used to treat other tissues as well? Yes, I believe so. So we have been treating them for dogs with neurological disease, and the results are pretty exciting. So I believe so, yeah.
All right. I think that's all the questions we have today. So thank you everyone for very insightful questions. All right, so we will be sending around the recording in the next few days, and so you'll have that for your records. And as I said, feel free to respond to that if you have any questions or if you are interested in any other aspects of JoVE. We're welcome to discuss those as well. So yeah, thank you, Amanda, for the excellent presentation, and thank you for all the attendees, and enjoy the rest of your day.
Thank
Yeah,
you
take
very
care.
much.
Yep. Take care, everyone. Bye bye.
Bye bye. Bye, everyone.